once organic matter has been catabolized and converted to this uniform two carbon chunks in the form of acetyl-CoA, it is fed into citric acid cycle, the roundabout of 8 chemical reactions which take place in the mitochondrial matrix, which purpose is to completely oxidize the acetyl group fed into this cycle to carbon dioxide and distill the reduction potential of this acetyl group in the form of reduced electron transport carriers, NADH and FADH2, which are then fed into mitochondrial electron transport chain to get completely oxidized on atmospheric oxygen and power with the released energy the system generating ATP. The 8 steps of tricarbonic acid cycle start with the condensation between end product of the cycle, oxaloacetate, a molecule which we have already encountered when we talked about gluconeogenesis, and acetyl group from acetyl-CoA to generate the 6 carbon compound citrate or citric acid. And the whole cycle of these chemical reactions takes its name due to its first intermediate citrate. Of note, citrate is a tricarbonic acid. It has 3 carboxy groups, and because of that, the citric acid cycle is also sometimes called tricarbonic acid cycle. One more synonym is Krebs cycle in the honor of Hans Krebs, who did major contribution to discover this roundabout of chemical reactions. During 8 steps of each cycle, the carbons, 2 carbons within the initial citrate intermediates are progressively chopped off in the form of CO2 in a series of oxidation steps. And at the end, these 8 steps return back oxaloacetate, which can be then used in the next round of the tricarbonic acid cycle, the citric acid cycle to oxidize 2 more carbons in the form of carbon dioxide and concomitantly release more reduced forms of electron transport carriers. In this way, during the rounds of the citric acid cycle, there is no net reduction or gain in the intermediates, and the flux through the citric acid cycle, the amount of the material which could be oxidized to a large extent can be regulated by the speed at which this circle, this roundabout of chemical reactions revolves. The citric acid cycle, the processes, chemical process within this citric acid cycle could be brought into 2 wide categories. The 4 steps constituting the oxidation steps per se, either dehydrogenization or oxidative decarboxylation, where actual oxidation occurs and the electron transport carriers are reduced and released, and 4 preparative steps which prepare the intermediates for the oxidation steps. If one would like to stage the citric acid cycle into different phases, then it makes sense to bring it down to two major phases. The oxidation phase, where the initial citrate is losing its two carbons within the backbone that at the end give rise to pure 4 carbon compound succinate. And the next phase, the regeneration of oxaloacetate, where this 4 carbon succinate intermediate is further oxidized and rearranged to give back the oxaloacetate. In the next part, we will discuss the details of each of the 8 steps of citric acid cycle and their purpose, bringing our description down to two phases: the oxidation of acetyl group into carbon dioxide and the phase of regeneration of oxaloacetate. The purpose of the first part of citric acid cycle, first 5 steps, is oxidation of the carbons within the acetyl group of acetyl-CoA into CO2 and concomitant release of the reducing power contained within acetyl group in the form of reduced form of electron transport carriers. So let's see how this is happening. The first step is the transfer of acetyl group, condensation of acetyl group within acetyl-CoA with the last step product of citric acid cycle, oxaloacetate, to form this tricarbonic acid citrate. As mentioned, from perspective of chemical logics, this step solves important problem of converting the methyl carbon, which is hard to oxidize, to methylene carbon, which is in principle possible to oxidize within the enzyme toolbox within mitochondria. The reaction having acetyl-CoA and oxaloacetate as substrates and producing citrate as a product concomitantly releases the reduced form of empty form of coenzyme A, deesterified form of coenzyme M back into the mitochondrial environment, making it ready for accepting next acetyl group through the PDH complex and fueling next cycles of TCA, citric acid cycle. And this reaction, release of acetyl-CoA, is deesterification reaction, where this highly energetic ester is hydrolyzed. And this fact renders this reaction extremely energetically favorable. It is almost irreversible reaction in a cellular condition. And this is important factor that drives citric acid cycle forward. The rate limiting compound intermediate in the whole citric acid cycle is oxaloacetate, which is present in very low concentrations. And without this reaction being highly energetically favorable, the whole citric acid cycle cannot proceed forward. The reaction is catalyzed by enzyme called citrate synthetase. Citrate synthetase is a dimer, and it is undergoing huge conformational change during the catalytic cycle, where important principle of enzymatic catalysis is induced fit. The first step of catalysis is activation of the enzyme by binding to oxaloacetate. Oxaloacetate induces huge conformational changes within enzyme, that it becomes more compact and generates binding site for acetyl-CoA. So at this step, within the catalytic center, so there are three critical residues, two histidine residues, and one aspartate residue, which bring together oxaloacetate, namely, to be more specific, it's electrophilic carbonyl group in a close proximity with acetyl group within acetyl-CoA, which is concomitantly gets deprotonated through aspartate residue converting to enol intermediate, and this methylene carbon within intermediate enol intermediate is turned into very efficient nucleophile, and this presence in a close proximity of the carbonyl group electrophile and the methylene group electrophile, nucleophile, allows the condensation between two carbons, which is from chemical perspective is called Claisen condensation. And this results in the formation of citric-CoA intermediate. So essentially citrate esterified, forming the thioester with coenzyme A. And this is again a very thioester is a very highly energetic compound which can release a lot of free energy upon hydrolysis, and that's what occurs at the final step. The hydrolysis of this thioester, which gives rise to final citrate product and coenzyme A, drives the backwards conformational change of the enzyme citrate synthase, converting it back to this less compact initial apostate, and this completes the synthesis of citrate. Next is the isomerization of citrate into isocitrate, and the idea, chemical logics of this reaction is to move this hydroxy group from tertiary alcohol position, which is very hard for oxidation, to the secondary alcohol, to the secondary position, to the isomer, citrate isomer with where alcohol in the secondary position, which is much more fit for subsequent oxidation. The reaction occurs through the intermediate called cis aconitate, and this is the unsaturated compound. So the reaction, which is catalyzed by enzyme called acotinase, is actually a reversible hydration-dehydration reaction, thus converting these two isomers to one another. The reaction is energetically unfavorable in at standard condition. However, in the conditions of normal cell, it is driven forward by pumping the citrates substrates through the previous highly energetically favorable step and evacuating the isocitrate product through the subsequent highly energetically favorable steps. So the interesting feature of aconitase is the fact that it has a iron sulfur cluster as in its catalytic center, and iron sulfur clusters are more typical for redox enzymes, such as ones that are found in the electron transport chain. And interesting fact that besides catalyzing this isomerization, the aconitase has also a moonlighting function in the iron homeostasis. In the case of when the iron is in shortage, so this enzyme loses this iron sulfur cluster and undergoes large conformational change, which turns it into regulatory factor regulating gene expression required for more efficient iron metabolism. The third step of a citric acid cycle is the first step where decarboxylation occurs. It is oxidative decarboxylation of citrate to alpha ketoglutarate. And in this step, one of the carbon inputs into citric acid cycle is released in the form of CO2. The chemical logics of this step is centered around oxidation of the secondary alcohol which was produced in the previous step into the carbonyl group with subsequent release, destabilization of this intermediate compound and release of CO2. Within this redox reaction, the reduced equivalents produced from the oxidation of alcohol into carbonyl group are evacuated in the form of reduced form of electron transport carrier, either NAD or NADP, respectively being reduced to NAD or NADH or NADPH. The preference of the cofactor is to a specific redox cofactor is dependent on the isozyme which is used for catalysis of these reactions, which we'll touch upon a little bit later. But the reaction itself occurs through the intermediate step which is trapped within the catalytic site of the enzyme catalyzing this reaction, isocitrate dehydrogenase, which is called oxalosuccinate. So oxalosuccinate is an unstable intermediate which is stabilized within the enzyme by manganese ion. And the manganese ion is also used to destabilize the carbon bonds within the oxaloacetate to kick out the carboxy group and convert oxaloacetate into this enol intermediate, which is highly unstable and immediately collapses to the final product, alpha ketoglutarate. So altogether, we started with 6 carbon compound and got a 5 carbon compound. This is the oxidative decarboxylation reaction in its nature, as mentioned, and this is a highly energetically favorable reaction. So it pulls the previous step, which was highly energetically unfavorable, forward. Fourth step is yet another oxidative decarboxylation reaction, and at the end of this step, another carbon input in the form of acetyl group is released out of the system in the form of CO2. This release is accompanied by evacuating the reductive power of the carbon within the alpha ketoglutarate in the form of again reduced form of a redox cofactor NAD. The output of this reaction is succinyl CoA, requiring respective input the empty upper form of coenzyme A. If you look closer at this reaction, all the inputs of this reaction, alpha ketoglutarate, coenzyme A, NAD, look very, very similar to the inputs of the pyruvate dehydrogenase reaction. And if you look closer at its main substrate, alpha-ketoglutarate, it looks very, very similar to pyruvate, except that the alpha-glutarate has this two additional carbon extension which pyruvate doesn't have. And not surprisingly, this reaction is catalyzed by enzyme alpha-glutarate dehydrogenase complex. The enzyme complex is extremely similar, almost identical to pyruvate dehydrogenase complex. So this is the example where of evolutionary divergence, which were very similar enzymatic machinery, can be repurposed for different functions. In fact, yes, it is very highly similar. In fact, very similar complex is also repurposed for oxidation of branched amino acids. So again, this is example of evolutionary divergence and repurposing. The net energetics of this reaction similar to the pyruvate decarboxylation is very, very highly energetically favorable reaction. At the end, we end up with the 4 carbon backbone, which is almost ready for regeneration of oxaloacetate. The step separating the product of previous step succinyl-CoA from the next phase, the regeneration of oxaloacetate, is deesterification or hydrolysis of succinyl-CoA into pure 4-carbon compound succinate. In fact, this is substrate level phosphorylation reaction. Its chemical logics is the conservation of energy derived from the hydrolysis of this ester bond in the form of phosphoanhydride bond generating the GTP, guanidine triphosphate, by phosphorylating GDP with an organic phosphate. In fact, GDP is analogous to ATP energy production, because GDP can be freely converted without energy loss to ATP by nucleoside diphosphate kinase. So it is the same as substrate level phosphorylation, giving rise to ATP. Concomitantly, this reaction also releases free form of coenzyme A, which was consumed in the previous step. From the energetic perspective, this reaction is almost neutral, and this is a balance between the energy released from hydrolysis of thioester and energy required for the formation of phosphoanhydride bond. The reaction is catalyzed by enzyme called succinyl-CoA synthetase, which is named due to the opposite reaction as compared to what occurs in the forward citric acid cycle. The catalysis of this reaction is a peculiar two-step substrate level phosphorylation reaction, and occurs in 3 phases. In the first phase, the succinyl-CoA undergoes transesterification, forming the carboxylic phosphatic anhydrate, in exchanging it to the in exchange to the thioester bond. The carboxylic phosphate anhydrate is then this energy of highly energetic energy of this highly energetic bond is used to phosphorylate a histidine residue within the active site of the enzyme, by a mechanism of substrate level phosphorylation, and then the histine residue uses energy of the phosphate bond to for the substrate level phosphorylation of GDP converting to GTP. And this concludes the first phase of oxidative phase of citric acid cycle. The purpose of the last step of citric acid cycle is regeneration of oxaloacetate, thus um, uh, completing the circle of the citric acid cycle roundabout. The purpose of these steps in essence to oxidize one of the methylene carbons within the succinate into carbonyl carbon, and this achieved in the three steps. In the first step, the succinate is dehydrogenized to derive the unsaturated compound fumarate. The reaction, which is catalyzed by enzyme succinate dehydrogenase, is essentially neutral at standard conditions, and the energetics of this reaction is a balance between the energy derived by the net oxidation of the middle carbons within succinate and energy required for evacuating or reducing, the electron transport carrier FAD to its reduced form of FAD H2, thus evacuating the reducing equivalents from the reaction. Interestingly, the succinate dehydrogenase is the only enzyme within the tricarbonic acid, the citric acid cycle, which is a membrane-bound enzyme. The others are soluble. The succinate dehydrogenase is a subunit of complex 2 of the electron transport chain, and this is a flower protein. It means that FAD is covalently bound to the context of enzyme, and within the complex 2, the electrons which are gained by reduction of FAD are directly fed into the electron transport chain, thus ready for production of ATP. An interesting fact is that the succinate dehydrogenase can be very efficiently competitively inhibited by a very similar compound, malonate, which is compared to succinate has one fewer the methylene carbons. And this is used as a pharmacological inhibition in pharmacological inhibition studies of the electron transport chain function, where you can efficiently block the flux of electrons through the complex 2. In next step, the fumarate is hydrated. The double bond is hydrated to turn it back into single bond, and this reaction occurs through this carbanion intermediate, which has a stereoisomeric form. And as a matter of fact, the fumarase enzyme which catalyzes this reaction and stabilizes this carbanion is a stereospecific enzyme. At the end, the malate form which fumarase produces is exclusively L isomer, which is determined by the arrangement of chemical bonds around this this chiral center, and fumarase never produces dimalate. The opposite is actually also true. The fumarase acts as a specific in isomer specific fashion, producing exclusively to fumarate this trans isomer of this dicarbonic acid, and never produces maleate, which is respective cis isomer. In the last step, the malate is oxidized once again to produce the end product of the citric acid cycle, oxaloacetate. In this reaction is catalyzed by malondehydrogenase, which we already encountered when we talked about the glyconeogenesis pathway and generation or transfer of NADH into cytoplasm to complete glyconeogenesis. This reaction is extremely highly energetically unfavorable in standard conditions. It is extremely highly endergonic, and it is for it, in order to proceed, it requires previous steps, highly exergonic steps of the citric acid cycle to pump the substrate on the left and constantly evacuate the produced oxaloacetate on the right through the first step of the citric acid cycle, the synthesis of citrate, which we remember is a very highly exergonic process. This way, the steady state concentration of oxaloacetate within the mitochondria extremely low, and it is a rate limiting metabolite in the metabolic flux through the citric acid cycle. So if we now look at what we actually gain by revolving the roundabout of citric acid cycle, if we have one acetyl input, then during the oxidation of acetyl group, the first phase of citric acid cycle, we convert the two carbons of the respective intermediates into carbon dioxide, which exit the system, and during the respective oxidative decarboxylation steps, we produce 2-NADH. At the deesterification step, which is coupled to substrate level phosphorylation, we produce 1 GTP, which is equivalent to 1 ATP, and in the following regeneration of oxaloacetate phase, we gain 1 FADH2 and 1 NADH, which is the final, which is a product of the final oxidation step of malate to oxaloacetate. So altogether, we can see that as energy production pathway per se, this is again an extremely inefficient pathway similar to glycolysis. If we count per glucose molecule, we would have 2 acetyl-CoA produced, and then at the end, per 2 revolutions of the cycle, which would require to oxidize these 2, we produce only 2 ATPs, which is the same as fermentative glycolysis. However, the power, the real ATP generating power of tricarbonic acid cycle and previous acetyl-CoA generating steps is in the production of these reduced equivalents, the reduced forms of the electron transport carrier. And as a result of these revolutions, these reduced forms are fed into oxidative phosphorylation system where they get completely oxidized on atmospheric oxygen, and this gives a lot of ATP. So one NADH, when fed into the electron transport chain, at the end gains approximately 2.5 ATP molecules, and one FADH2 which is fed through the complex 2 of the electron transport chain, gives a little bit less 1.5 ATP molecules. So now we can look what we gain in terms of ATP when we oxidize completely one glucose molecule through the cellular respiration pathway, including the glycolytic steps, the generation of acetyl-CoA, and the oxidation of acetyl group in the citric acid cycle. If we count per one glucose molecule, then in the first part, this red boxed one, the glycolytic steps, we get, as we know, 2 ATPs in total, which is the 4 ATPs in pair phase subtracted by 2 ATPs in the preparative phase. We also get 2 NADH, which are evacuated during the glycolytic pathway, and this NADH at the end is fed into mitochondria and electron transport chain, and depending on the way how it's fed, as we will see later, one could get for these two NADH either 3 or 5 ATP molecules. The generation of acetyl-CoA gives us 2 NADH, hence 5 ATP molecules. And the steps of the citric acid cycle will give us 6 NADH in total per 2 acetyl CoAs generated per glucose molecule. So hence we get 15 ATPs generated through the oxidative phosphorylation. Then we get purely 2 ATPs and gain 3 ATPs, 1.5 per each FADH2, feeding the electron transport chain through the succinate dehydrogenase. And if we add up what we can gain from one glucose molecules when we oxidize it through the cellular respiration, we get from 30 to 32 molecules of ATP. And if we just try to consider its efficiency of this energy conservation, as we discussed, and at the standard conditions, the burning of glucose with oxygen would produce approximately around 3000 kilojoule per mole of energy. But if we would count how much energy we produce in standard conditions when we would hydrolyze ATP, the end product of this burning within this complex chain of enzymatic reaction, we get approximately 1000 kilojoules per mole in standard conditions, which is approximately 30, like approximately 1/3 efficiency. But if you would recalculate and factor in the actual concentration of metabolites and oxygen concentration, etc., what happens in real conditions, so we would see that this pathway conserves approximately 65% of chemical energy released by burning of glucose and oxygen in the form of the energy in ATP. So if you go back to our internal combustion engine analogy, the car engines would conserve or convert approximately only 20% of energy of burning of fossil fuels with oxygen into the useful form, the form of locomotion. And the cellular respiration conserves more than 50% of chemical energy produced by burning of the organic food with atmospheric oxygen, which underscores the efficiency of the energy conservation, which is uh, achieved by a naturally evolving mechanism of um, cellular respiration.